Why have so many Alzheimer's drugs failed despite decades of research? And why have breakthroughs in understanding its fundamental mechanisms remained so elusive? For decades, while the primary focus has long been the amyloid-beta hypothesis, the high rate of clinical trial failures has prompted the scientific community to conduct a necessary reevaluation of the disease's core drivers. Tau pathology, especially the formation of tangles inside neurons, is now discovered as much more closely linked to cognitive decline. However, moving toward tau presents a fundamental biological challenge. In humans, tau is not a single protein, but a balanced family of 6 distinct isoforms, 3 known as 3R and 3 as 4R. Critically, Alzheimer's disease is a mixed taupathy, where the toxic aggregation involves both the 3R and 4R types. Therefore, an effective therapeutic strategy requires a clear understanding of this complex, human-specific interplay. To accurately investigate this complex tau pathology, researchers depend on animal models. These models are foundational, but conventional transgenic models are built on a methodology with inherent and critical limitations. This conventional approach relies on the random integration of a gene, which limits its capacity for accurately modeling the precise human tau isoform landscape. Crucially, the endogenous mouse tau gene itself does not produce the same 6 isoform balance as the human gene, creating a foundational biological mismatch. Furthermore, the use of non-native artificial promoters leads to unnatural and variable overexpression, which does not reflect the carefully regulated physiological levels in the human brain. This can cause a cascade of issues, most notably phenotypic drift, where results become inconsistent across different cohorts or generations. The model is, by its design, an incomplete system. This leads directly to the critical consequence, a high risk of failure in clinical translation. When a preclinical model cannot accurately recapitulate the specific human pathology, the drugs developed using it are destined to struggle in human trials. To address these limitations, a radically different approach to model generation is required. The solution is to re-engineer the model's genetic foundation using humanized genomic ortholog replacement. This is not a typical transgenic approach. Instead of randomly adding a gene, this method uses large fragment gene replacement to precisely swap the entire mouse MAPT gene locus with the full-length human MAPT gene. This is the core of Cyagen's Hugo GT technology platform. By placing the human gene under the control of its own endogenous promoter, the model successfully expresses all 6 tau isoforms at physiological levels. This design effectively avoids the artifacts of random integration and overexpression, providing a genetically stable and biologically relevant platform. The H-Tau series is a comprehensive platform for studying tau pathogenesis. It contains 3 humanized models, a wild-type isogenic control, the P301L mutant, and the P301S mutant. These mutants allow us to compare different disease timelines. To verify the degree of humanization and confirm that the models perform as expected, we conducted extensive validation studies. Firstly, HT7 antibody staining confirmed human-specific tau protein expression. Furthermore, staining with the AT8 antibody, which recognizes a key pathological phosphorylation site, revealed dense intraneuronal tau aggregates in the hippocampus of the mutant mice. This molecular pathology is directly linked to the behavioral results. On the behavioral side, a comprehensive evaluation using the open field, rotorod, and novel object recognition tests revealed a progressive, mutation-specific cognitive decline. Specifically, in the novel object recognition test, the aggressive P301S model showed a deficit by 6 months, while the P301L model followed by 9 months. This staggered progression is crucial. It allows researchers to model distinct stages of the disease and define clear windows for intervention studies. The utility of this humanized platform extends directly to therapeutic and diagnostic development. Because these models express the complete physiological set of human tau isoforms, they are highly relevant for evaluating the efficacy and specificity of next-generation therapeutics. For example, the development of immunotherapies requires a system where antibodies can recognize human-specific epitopes, a function only a humanized model can faithfully provide. Similarly, The testing of antisense oligonucleotides, ASO, demands a model with the native human mapped gene sequence to ensure accurate target engagement. Furthermore, the validation of novel PET tracers depends on a system that presents the full range of human tau targets across different stages of aggregation. The H-tau series, therefore, offers a significantly enhanced translational link, facilitating the necessary connection between rigorous preclinical research and potential human clinical success. We believe better models make better science. To review the full validation data and learn more about the diverse applications of these humanized models, you can find the link to the www.cyagen.com in the description below.
Cyagen help you discover life.